Good morning to our respective lecturer, Dr. Professor Dr. Ismail Yusuf. It is nice to be here. I'd like to start by introducing my groupmate and myself. My name is Noraina Zulaika Najwa binti Merian, and my groupmate is Rasharani Anajani and Gloria Willy. Today, I'm going to talk about the effect of different type of cutting with and without Auxin on the yearly growth of Hibiscus rosa-sinensis. Before we talking deeper in this topic, I like to I like to show you what we focus on. We want to let you know that our research focusing to the yearly growth of different types of cutting of Hibiscus rosa-sinensis with the presence of hormone or not. Then we want to focus on the cofactor, which is maybe by the hormone of the type of cutting or the type of cutting affect the yearly growth of Hibiscus rosa-sinensis L. Third, the environmental factor, one of the part that essential in the growth developmental of Hibiscus rosa-sinensis L. The last one is we want to focusing does different cutting affect the growth of Hibiscus rosa-sinensis because different cutting show the different maturity in cell. After that, I want to talk about our research background. Hibiscus rosa-sinensis, as we know, is a national flower of Malaysia because it has a five petal. Five petal represent of five principle of Rukun Negara, our national principle. Then the red color is show the Malaysia's perfection. Then Hibiscus rosa-sinensis L from Malvasia family and the tribe is Hibiscae. It is widely planted in Malaysia as an ornamental plant, and it is widely known as the ornamental plant in subtropic and tropics. In this research, we will apply hardwood, semi-hardwood, and softwood cutting on by applying the stem cutting using the stem of the Hibiscus rosa-sinensis L. The stem cutting, as we know, it is the process that applied to rooting a piece of plant whether at least one knot to rotate on the growth medium such as moist soil. And as we know today, it is very popular that stem cutting is one of the vegetative propagation which very inexpensive and easy to apply. Then I want to proceed our presentation to my groupmate, Rachel Rani Najani. Sorry, uh, I want to proceed my presentation. How we want to detect the Hibiscus rosa-sinensis. Hibiscus rosa-sinensis, we can detect it by its leaf, which is it have glossy leaf and pinate vein, also oval shape with pointed tip. Then we can see there are five petals of the red Hibiscus rosa-sinensis. But as we know, there is popular hibiscus which in white, pink, yellow, and the others. But we want to focus on the red Hibiscus rosa-sinensis, which is very popular in tropic and subtropic. And we can see the anthers of the Hibiscus rosa-sinensis, which is yellow, or sometimes it is in orange. In orange, at the tip of column. So I want to proceed our presentation with our group head, which is Rachel Rani and Ajani.
Our objectives for this research is to study the cofactor of Hibiscus rosa-sinensis L, which contributes to easy-to-root characteristics in cultivars without artificial hormones such as IAA, IBA, and etc. Second, we wish to study the effect of auxin hormone on the rooting initiation and formation of Hibiscus rosa-sinensis L. We also want to study the effect of different types of cutting on the early growth of Hibiscus rosa-sinensis L. And lastly, we want to study the influence of temperature, humidity, and environmental factors on the early growth of Hibiscus rosa-sinensis L. Our hypotheses for this research are the type of wood cutting, which will influence the early growth of Hibiscus cultivars. Softwood cutting will have the slowest root formation due to its leaf being trimmed, since leaf purpose are to create food through photosynthesis process. Next, semi-hardwood cutting will have intermediate root formation, but it is more liable to rot and could desiccate quickly. Lastly, hardwood cutting is the most easily to root due to its cells being mature. Our second hypothesis is that hormone will have influence on the early growth of Hibiscus rosa-sinensis. Rooting hormones auxin, such as ABA and IAA, should improve the growth of the roots. However, high level of auxin will inhibit root formation instead of enhancing them due to the cofactors present in the cultivars. Lastly, our hypothesis is that environmental factors also affect the early growth. Hibiscus plants are adapted to hot climate and they grow on sandy soils. Medium temperatures, which are the optimum one, that ranges from 26 degrees Celsius to 30 degrees Celsius, will influence the time of the hibiscus plant to initiate rooting. Any temperature above 34 degrees Celsius, the root of the hibiscus cultivar will become more filamentous and weaker. Other factors that are on optimum level, such as humidity, soil pH, and others, may enhance the early growth of cultivars. I will now pass the presentation to Gloria Willy.
So for literature review, we divided it into two parts, which are vegetative propagation and cutting method. So for vegetative propagation, it offers the opportunity rapidly to overcome the limitations in agricultural crops. These limitations mentioned are, for example, the long generation time of plant, irregularity for fruiting and flowering, and also high incidence of outbreeding. So these limitations can be overcome by circumventing the need for sexual reproduction and also facilitating the capture of individual genotype. So next is for cutting method. Cutting method is also known as any vegetative plant part which, when detached from its parent plant, is capable of regenerating the mission organ or organs. So for cutting method, it is mainly used to develop plant similar to its parent plant, and it is also physiological, biochemical, and cytological influence the root development. So root development in cutting method usually influenced by the species, depends on the species, and also among plant which we chose to be cloned. So two major groups of stem cutting are leafy softwood cuttings from relatively unlignified young shoots, and also leafless hardwood cuttings from older and more lignified shoots. So these are the materials that we use for cutting, which are Hibiscus rosa-sinensis, sharp cutting utensils, peat moss potting soil, rooting hormone, sand, clay, earth, water, cups, and ruler. So for the methods, these are the basic methods for cutting, where we again divided into three categories, three parts, which are for softwood, semi-hardwood, and hardwood. So we remove them from the parent plant using sharp cutting utensils, and then we remove all the flowers and flower buds from the cuttings. And using a ruler, we measure each branch length and record the measurement on the data sheet. Next, we proceed to cutting approximately one of an inch below the bud using sharp cutting utensils. And then we dip each of the 5 from each of category, from softwood, semi-hardwood, and hardwood, into rooting hormone. Rooting hormone is function to enhance the rooting of the plant. And then we place 10 cuttings in each of 4 fill containers where we put them 3 inches away from each other. And then each day, we will water each cutting using quarter of cup of full water, where we make sure that it is not being flooded in the media. Then the planted cuttings are measured each once a week, and we take note on how much it has grown, and then we compare to its original height, and we determine the percent it has grown in a recorded data. And then lastly, for the at the end of 4-week time period, uproot each specimen and determine the number of successfully rooted plants in the 4 different media.
The work schedule that we plan for this research is that for phase 1, especially for research and proposal, it will be covered from week 1 until half of week 2. For phase 2, which is the pre-experiment, in pre-experiment, we wish to collect materials and prepare before we done the real experiment. And phase 2 will be covered from week 2 until week 3. For phase 3, which is the experiment, it will be covered from week 3 until week 7. And lastly, for phase 4, which is the post-experiment, that includes the observation and recording of data, report writing, and presentation, will be covered in week 8.
So these are the expected outcome that we have discussed, where again, we divided them into 3 parts for softwood, semi-hardwood, and hardwood. So for softwood, with hormone and without hormone, both where with leaf, they will produce more number of roots compared to without leaf. And then for semi-hardwood, with hormone and without hormone, also the same. With leaf, they will produce more number of roots compared to without leaf. And lastly, for hardwood, with hormone and hardwood without hormone, will also produce the same outcome, which is with leaf, again, produce more number of roots compared to without leaf. So, in conclusion, we hope that our research procedure will go smoothly as planned, and we will also get a result approximately based on our expected outcome. So that's all for our presentation. Thank you.